But how was teosinte transformed into maize? Back in Dr. Dobley's lab in Wisconsin, I learned about the genetic changes involved. One of the main differences between teosinte and maize is that the teosinte seeds are encased in this really hard fruit case that makes it really difficult to eat. So clearly that's something that had to change.
That's right, and the remarkable thing is that having a fruit case versus not having a fruit case is basically controlled by a single gene.
A single gene?
A single gene.
To test this gene's function, Dr. Dobley's team did a clever experiment. They carefully crossbred maize and teosinte to introduce the maize version of the fruitcase gene into teosinte plants. When they did that, the teosinte kernels, which are normally enclosed in a hard fruit case, became partially exposed, almost like little corn kernels. When they did the opposite, putting the teosinte fruitcase gene into maize plants, the fruitcase became larger and started to cover up the maize kernels, similar to teosinte.
One gene makes a pretty dramatic change.
So another really obvious difference between teosinte and corn is that teosinte produces dozens of these little tiny ears on a plant that branches a lot, and corn just produces a couple of ears on a plant that hardly branches at all. So what's going on there?
There is one gene that we've identified that plays a central role in that process, and you could call it the branching gene.
Dr. Dobley explained how putting the teosinte version of the branching gene into maize made the maize plants more branched, like teosinte. And putting the maize version of the gene into teosinte made the teosinte plants less branched. Dr. Dobley has shown that the fruitcase gene, the branching gene, and just a few others, a small number of genes, just as George Beadle predicted, were responsible for setting in motion all the major differences between maize and teosinte. But how could so few genes cause such huge changes? Why were these genes so powerful?
They both belong to a special class of genes called regulatory genes, and these are genes that directly regulate the activities of other genes.
And so when we move the teosinte version of one of these genes into a corn plant or vice versa, we're actually changing more than just that one gene.
That's right, they can turn other genes on and off. You could think of these genes as something like the conductor of an orchestra. And if you would take the conductor from one orchestra and give that orchestra, say, a new conductor.
Just like we did moving some genes from teosinte to maize or vice versa.
Right, and you could get a very different quality of music, even though all of the musicians and all the instruments remain the same.
These regulatory genes probably influence the activity of hundreds of other genes, which explains how mutations in just a few regulatory genes could dramatically transform teosinte.